Hey everyone, Mr. Larson here. So far we have talked about basically DNA replication, how it goes from one strand of DNA to two strands of DNA. We've talked about transcription, which is basically taking that DNA and turning it into what is called RNA. And we've talked about RNA and what RNA is and so on. So now we have created a DNA molecule or DNA strand. Our DNA strand has now become RNA. That RNA has now been transcribed and is now going to move on to this next part, which is called translation. So translation basically takes the messenger RNA, that mRNA that's been created from DNA, and is now going to create proteins, or what is called protein synthesis. So proteins are synthesized on ribosomes, where the mRNA is translated into a sequence of amino acids. So if you recall back in your biomolecule lesson, that amino acids are the basic structure of a protein. So proteins are made up of exclusively amino acids. So the mRNA basically brings in all these amino acids to create this protein. So how does translation work? Here we're going to investigate that. First, what we need to know is that each amino acid that we see, that we know of, has a specific mRNA sequence associated with it. So here we have a genetic code wheel. This will help us understand where and how we sort of figure out what amino acids are coming in. So you will always start in the middle and work your way out. So let's say we have a mRNA sequence that is UCA. So our mRNA sequence is UCA. That means a serine amino acid is going to come and bind to that different that mRNA strand. That is where it's going to bind. So basically, you just start from the middle. So let's say the next sequence is GAG. So GAG is glutamic acid. So the next amino acid is going to bind is that amino acid, glutamic acid. So how do we know this is how it works? These are what are called codons. A codon is a sequence of 3 nucleotides that code a specific amino acid. So it's always 3. Always 3. There are only 3 nucleotides for a codon. No more, no less, only 3. So let's say we have AUG. So AUG codes for what is called methionine. If we go back to our genetic wheel here, we go to AUG. Look at that, methionine, just like what it says here. So UUA codes for leucine. So let's go back to our wheel. UUA, leucine, right there on the end of the wheel. So that's how we know that is the amino acid that binds to the mRNA in the ribosome. So an anticodon is a sequence of 3 unpaired bases attached to the transfer RNA that corresponds to each mRNA codon. So the anticodon is basically the second side of an mRNA. So AUG would be UAC. So A would bind to U would bind to A, G would bind to C. So basically, a codon is basically the sequence. Anticodon is the opposite of that, or is basically what pairs to the codon. So in translation, we use both codons and anticodons. So here, mRNA has AUG and relies upon transfer RNA to bring over the amino acid in the ribosome. So here in the blue, this is our ribosome. Our mRNA runs through the ribosome. The transfer RNA brings over the amino acid, and that is what creates the protein. This is the entire process of translation. So translation basically means that you are translating the mRNA to create proteins. So the polypeptide assembly line is basically what is formed through the ribosome. So you basically taking all of those amino acids, basically combining them together, and that is basically what forms that polypeptide chain, and that polypeptide chain forms together to create a protein. So that is it for translation. You're also going to watch a video here in a little bit about how this whole process works in sort of a 3D model. So I hope you guys enjoy.